ELISA, which stands for Enzyme-Linked Immunosorbant Assay, is a test used to determine the presence of antibodies in an individual. If an individual has been infected with something foreign to the body, like a virus, bacteria, or a parasite which contains antigens, the body will make antibodies against that specific antigen, and an ELISA test can be used to detect these antibodies. So for example, with the current coronavirus or SARS-CoV-2 pandemic, an individual who has been infected mounts an immune response and produces antibodies specific to SARS-CoV-2 for about 2 weeks, during which time they could be tested with an ELISA. While you would not be able to use this test to see if you have recently been infected, this test will be useful for individuals who are experiencing symptoms of the virus. Before we demonstrate our SARS-CoV-2 ELISA test, we will first explain how an ELISA works. An ELISA plate is covered with the antigen of interest, and in this case, the antigen would be particles from SARS-CoV-2, which could be any of the proteins on the surface of the virus. It's important to note that the plate does not contain the virus itself, but proteins, which are the antigens to which specific antibodies are synthesized. A clinician collects fluid from the human body known to contain antibodies, which is usually blood. The red blood cells are removed so that you will have only the clear serum left, which contains the antibodies. The serum samples are added to the wells of the ELISA plate, and you can see that in this example, sample 1 contains antibodies specific to SARS-CoV-2, and sample 2 contains only nonspecific antibodies. The specific antibodies bind to the antigens on the ELISA plate. Then the wells are washed to remove any nonspecific antibodies. We cannot see these antibodies yet, so we need to perform a few more steps to be able to detect them. Next, we add the secondary antibodies specific to the primary anti-SARS-CoV-2 antibodies. The secondary antibodies each have an enzyme attached to them which can react with something else to result in a color change that we will be able to see. After 5 minutes, the secondary antibodies are bound to the primary antibodies, and we wash the wells again to remove any unbound secondary antibodies. You can see that there are no secondary antibodies in the well containing sample 2 because there were no anti-SARS-CoV-2 antibodies for them to bind to. Lastly, we add the substrate, which binds to the enzymes on the secondary antibodies. The enzyme and substrate undergo a reaction in which the enzyme converts the substrate into a visible colored product. Now we can see with our eyes which sample contained the antibodies specific to SARS-CoV-2, and we know that sample 1 tests positive for SARS-CoV-2 and sample 2 tests negative for the virus. Now we'll show you what an ELISA test actually looks like in the lab. ELISA plates usually have 96 wells like this one, but we're going to do our test in a partial ELISA plate with fewer wells since we don't need that many. We have 3 tubes containing serum samples with unknown antibodies, and we want to know if they contain SARS-CoV-2 antibodies. We also have a positive control solution which contains SARS-CoV-2 antibodies, a negative control solution which contains no antibodies, secondary antibodies, and a color reagent, which acts as the substrate which reacts with the enzymes on the secondary antibodies to produce that visible color change. We are going to add the positive and negative controls as well as the 3 samples, each to 3 wells of the ELISA plate. This is called performing the ELISA in triplicate to make sure our results are accurate. You can see that we skip a row of wells between the positive control, negative control, and the samples to prevent cross-contamination and make it easier to see what we're doing. We add 2 to 3 drops of each solution to the wells to make sure that the bottom of the well is coated with solution. When the samples and controls have all been added, we incubate them for 5 minutes to give the SARS-CoV-2 antibodies time to bind to the antigens on the ELISA plate. After 5 minutes of incubation, we will wash the wells with ELISA wash solution, which is a mild soap and low concentration salt solution. This gets rid of any nonspecific antibodies in the wells, leaving the SARS-CoV-2 antibodies bound to the antigens on the ELISA plate. Make sure you empty the wells into the sink, tipping the plate in the direction of the same solutions so that the different solutions do not mix with each other. The washing step is repeated 3 times to make sure all nonspecific antibodies are washed out. After washing, tap the ELISA plate face down on a paper towel to get rid of excess liquid. Next, we add 2 to 3 drops of the secondary antibodies to each well that had a control or a sample in it. As we saw in the ELISA illustration, the secondary antibodies will bind to any SARS-CoV-2 antibodies that are on the ELISA plate. When all the wells have secondary antibodies, we incubate for another 5 minutes to let them bind. After 5 minutes of incubation with the secondary antibodies, we wash the ELISA plate with the ELISA wash solution 3 times just like before to remove any unbound secondary antibodies. The final step is to add the color reagent, which will react with the enzyme that is bound to the secondary antibodies. As we saw in the ELISA illustration, this reaction will only occur in wells which contain SARS-CoV-2 antibodies. The result will be a visible blue color in the wells positive for SARS-CoV-2. You can immediately see this color in the positive control and in 2 of the samples, but we will give the wells time to develop to see a stronger color. After a few minutes, it's easy to see that the positive control, sample 2 and sample 3, test positive for SARS-CoV-2, while the negative control and sample 1 are negative. Thanks for watching, and we hope you learned a lot today.